Good afternoon in Philadelphia from New Jersey. I thank you for the opportunity to present on behalf of the Protein Data Bank, which, as you see, is celebrating its 50th anniversary. I'll talk about sickle cell disease in 3D and specifically introduce hemoglobin S, and then a drug that can be used to treat sickle cell disease. And then I'll outline some of the possible treatments using genetic manipulation to actually cure sickle cell disease. So here's a brief outline of the presentation. I'll first talk about the Protein Data Bank, touch on our efforts supporting global research and education and our efforts for the pandemic. And then I'll talk about sickle cell disease and conclude with acknowledgements. And I'll also advertise some job openings that may be of interest to members of the audience. So the Protein Data Bank was established in 1971 as the first open access digital data resource in all of biology. At that time, there were just 7 proteins. It's grown enormously, and now there are more than 175,000 structures of proteins, DNA and RNA and their complexes with one another and with drugs, etc. It is the single global archive for experimental structure data. It's currently managed by a global international consortium known as the Worldwide Protein Data Bank, or WWPDB, which consists of data centers in the US, Europe, and Japan, and a specialized data center known as the Biomag ResBank, which houses information generated by NMR spectroscopists. In the right panel of the slide, you'll see some of the structures that actually inspired the launch of the Protein Data Bank, and hemoglobin in the top right will figure prominently in the stories that I'm going to tell you today. The US funded WWPDB data center is known as the RCSB Protein Data Bank. We support research and education around the world. We principally support the data generators, the data depositors, the structural biologists, and to date, about 50,000 people have put structures into the PDB. We deliver this information to many millions of data consumers around the world using our research-focused website, rcsb.org. And last year, we delivered didactic materials and PDB data to educators and students around the world through our pdb101.rcsb.org, reaching more than 800,000 individuals last year. We're jointly funded by the National Science Foundation, various national institutes of health, and the US Department of Energy. And you can see on the right that this is a very big team effort. The picture that was taken here celebrated the release of structure number 150,000 by a team that operates at Rutgers University in Piscataway next to New Brunswick here in New Jersey, out of the San Diego Supercomputer Center at UCSD, and out of an institute at the University of California, San Francisco. We turn global data into global knowledge. Literally every day of the year, we're receiving information, 3D structure information from structural biologists working on all inhabited continents. We process this information in collaboration with our Worldwide Protein Data Bank partners, each regional data center taking responsibility for local depositions. The output of the deposition and biocuration team goes at the RCSB Protein Data Bank here in the US to an archive management and access team, which is responsible for consolidating the newly arrived information, integrating it with a series of external data resources, and then making it available through the archive, the green oil drum in the middle of the picture, to more than 400 different external scientific data resources, many of which repackage and redistribute PDB data. We distribute the contents of the Protein Data Bank archive through our website, rcsb.org, which is operated by the data exploration team, and through our educational and outreach website, known as pdb101.rcsb.org, an introductory version of the Protein Data Bank, which is operated by our outreach and education team. And it's the concerted effort of these four services that allow us to convert global data into global knowledge for millions of people around the world. Before I talk about sickle cell disease, I want to say something about our response to the pandemic and explain to you the pretty dramatic effect that structural biologists have had on our understanding of SARS-CoV-2, the cause of the current pandemic, and explain how we've been working with structural biologists, data generators around the world to make this information freely available to researchers everywhere who are intent on combating the pandemic. So you know the timeline. In late 2019, an unexplained influenza-like illness was observed in Wuhan in the People's Republic of China. And in January, the causative agent of this new viral infection was identified as SARS-CoV-2, a novel coronavirus, very closely related to SARS-CoV-1, which struck about 18 years ago. The full genome sequence of the virus was released on the 10th of January, which provided information about all the proteins that make up the virus and the proteins that are used during viral infection. In February of 2020, structures of viral proteins literally began pouring into the PDB. The very first of these I'll show you in a subsequent slide was that of non-structural protein 5, or the main protease. As of today, there are approximately 1,180 PDB structures. I'm sorry, this slide is slightly out of date, of SARS-CoV-2 proteins. The virus has been referred to as the unseen enemy, but we have a molecular artist on our team who is based out in San Diego, in La Jolla at the Scripps Research Institute, David Goodsell. And David has been painting portraits of the virus. And I find this one particularly compelling. It is a picture of the virus shown. Let me put the laser pointer on. It's a picture of the virus inside a respiratory droplet. This is the border of the droplet. This is air. This is water. Here's David's rendition of the virus, and it's in this droplet that would be coughed up by somebody who's infected with the virus. And it's this droplet which is the unseen enemy. This is the mode of transmission of SARS-CoV-2, and the reason that it has spread around the world, been spread around the world by individuals who are, upon initial infection, largely asymptomatic, but nonetheless very infectious to people around standing in the vicinity. So it's these droplets that mean that we have to wear masks and we have to practice social distancing until we get enough people vaccinated to tamp down the pandemic. Our efforts have been focused on prioritizing all the incoming new structures, helping our depositors make those available to the scientific community immediately, consistently biocurating, annotating these structures, adding value to the information, and then releasing them along with related resources. If you go to our rcsb.org website, you'll see this logo, this graphic in the top half of the homepage, and you can click on that and get to all of our coronavirus resources. One of which is this very popular video about hand washing, and it explains how the soap molecules, the fatty acids that are in soaps, are capable of denaturing the virus and inactivating it and rendering it non-infectious. Many of the materials that we've generated over the past year and a bit have been used extensively. This particular video has been viewed nearly 500,000 times and counting. So as I said, our efforts have been in the pandemic in supporting all of the structural biologists around the world who are who've been working on trying to understand the virus through structural studies of SARS-CoV-2 proteins. Our efforts have been focused on getting the data into the PDB as quickly as possible, expertly validating it and biocurating it, and then getting it out to the public as quickly as possible with all of the appropriate annotations associated with each structure. Shown on the right is an all-atom representation. Each one of these little spheres corresponds to a different non-hydrogen atom. This is an all-atom representation of non-structural protein 5, otherwise known as the main protease. It consists of two identical subunits, one in dark blue, one in light blue. Each of these subunits is an enzyme that's responsible for cutting up the viral polyprotein, which is made upon infection of the host cell by the virus, which releases a preformed messenger RNA into the cytoplasm, which is then picked up by the host ribosome, and proteins are made. It's this protease and another one known as the papain-like protease that are responsible for cutting up the polyprotein and liberating all the different parts of the viral toolkit that are used during the infection. Inhibition of this protease or the papain-like protease would be sufficient to stop the infection. And what you can see here in red is the 3D structure of a bound inhibitor. This is sitting in the active site, blocking it, rendering the enzyme inactive. You see on this face of the dimer, the inhibitor is very easy to see. And on the opposite face of the molecule, the light blue subunit, you can see just this atom is just peeking out on this right-hand side. Every structure in the protein data bank has a unique identifier. In this particular case, it's 6 Lima uniform 7 using the NATO alphabet. This is a drug discovery target. Pfizer has 2 phase 1 clinical trials in operation right now, one for an intravenously delivered molecule and one for an orally delivered molecule. And it's clear from some of the press releases from the company that their goal is to get an expedited emergency use authorization for the pill form of the inhibitor of the protease out to the public before the end of this calendar year. The next slide here depicts the structure of the RNA-dependent RNA polymerase. This is a complex of 1 copy of non-structural protein 7, 2 copies of non-structural protein 8, and 1 copy of non-structural protein 12. The light blue here is non-structural protein 12. This is actually the enzyme that generates an RNA copy of the RNA template. In addition, there's this light blue, purple rather, molecule, which is the single copy of NSP7. And then you see we have these 2 copies of non-structural protein 8, which act like molecular chopsticks, holding the RNA duplex here so that it doesn't escape from the enzyme. This is a validated drug target. The US FDA approved remdesivir molecule, which was repurposed from another drug discovery campaign, received emergency use and then subsequently full authorization. And the binding site of remdesivir is right in here. And when remdesivir binds, it blocks the action of it gets incorporated into the RNA and then blocks the action of the RNA-dependent RNA polymerase. This is also enough to stop the virus in its tracks. The problem with remdesivir is that it doesn't bind very tightly to this particular enzyme. It wasn't designed for SARS-CoV-2. I think it was designed for Ebola. And as a result, it's not a particularly effective drug. I have much higher hopes for the Pfizer main protease inhibitor, which is currently in phase 1 clinical trials. And then many of you have seen renditions of this protein on the TV news. This is the spike protein from the virus. It's a trimer consisting of a total of three identical subunits that may be conformationally different, even though their amino acid sequences are exactly the same. And it's this spike protein that binds to the angiotensin converting 2 enzyme on the surface of human cells and allows the virus to gain entry. It's a vaccine discovery target. The mRNA vaccines actually encode this protein, as do, so the 2 mRNA vaccines, the Pfizer and the Moderna, have inside them an mRNA which encodes this protein. The Johnson & Johnson and AstraZeneca vaccines have a piece of DNA which encodes for this same protein. This is also the target for antibody discovery, monoclonal antibody discovery for passive immunization of individuals. And if you recall that when the former President Trump was hospitalized for COVID, he received one of these monoclonal antibodies. I think it was the one from Regeneron. He also received remdesivir. And so that ends what I wanted to say about COVID. As I said, there are now nearly 1,200 structures of SARS-CoV-2 proteins in the protein data bank. They are contributing to our fundamental understanding of the virus, and they are also contributing to vaccine design efforts, monoclonal antibody design efforts, and drug design efforts around the world, with the goal, of course, of combating the pandemic. Now, let me change topics and focus on the story of sickle cell disease, and I'll start with a brief historical introduction, because this actually was the very first molecular disease to be defined in the scientific literature. In 1910, a clinician in Chicago noted when looking at a peripheral blood smear of an individual with severe anemia, that this would be a normally shaped red blood cell. Some of the red blood cells had this very abnormal curved shape, sickle shape, so this was the first indication that the severe anemia that we know to be associated with sickle cell disease causes a structural change in the red blood cell. In the 1920s, it was determined that this is an autosomal recessive disease in which both parents have to carry the trait, and then there's a 25% chance that the progeny will be affected by the disease, having inherited both copies of the defective gene, whereas there's a 25% chance that the child will not have inherited the defective gene, and a 50% chance that the child will have inherited one copy of the defective gene, meaning that they have what's called sickle trait. They're carriers of the gene, and unlikely to be affected by a sickling crisis, except in very, very rare situations of low oxygen. In the 1940s, it was determined that it was the deoxygenated form of the oxygen-carrying molecule hemoglobin that was responsible for the sickling. So you see this light micrograph, black and white light micrograph here, showing normal shaped red blood cells that have been exposed to blood, they're in blood that's been exposed to air. The right-hand panel shows abnormally shaped red blood cells that come from blood that was freshly drawn from an individual with sickle cell disease and then sealed so that oxygen couldn't get in. So this is a reversible process. And then finally, in 1949, Linus Pauling, who should be known to many of you as a brilliant American chemist who went on to win the Nobel Prize, became a somewhat quirky proponent of vitamin C as a cure-all. Earlier in his career, he, in 1949 specifically, using protein electrophoresis, he and his colleagues were able to determine that there were discrete differences in the proteins of individuals that had no evidence of sickle cell disease versus those who had sickle cell anemia. When they looked at individuals who were the unaffected siblings of individuals with sickle cell disease, they saw two peaks in the electrophoretic pattern of the proteins, indicating that it was a mixture of normal and abnormal hemoglobin, and they were able, making a 50-50 mixture of these two, of this kind of a sample, to replicate what they saw with the sickle cell trait. So it was this information that allowed Linus Pauling and his colleagues to document that sickle cell disease was in fact a disease of a genetic disease that had a molecular explanation at the level of human protein. So before I actually introduce the mutation that's responsible for sickle cell disease, let me just orient you to the 3D structure of hemoglobin. Now, in most of us, our red blood cells are carrying what is known as hemoglobin A, which is this is a tetrameric molecule consisting of 4 subunits, 2 alpha chains and 2 beta chains. And you can see on the right-hand side the structure of the deoxy form of the enzyme of the oxygen carrier here, and then in the lower half of the slide you can see the 3D structure of the oxy form of the hemoglobin tetramer. So this is the form of oxygen that you see in the lungs as it's being loaded, the form of hemoglobin in the lungs that is loaded with oxygen. And this is the form of hemoglobin that one finds in veins and in capillaries, where the oxygen has actually been given up to the surrounding tissues. The two structures are different. You can see that, I think, clearly from these two discrete views, but this is even more evident in this animation that's alternating between the oxy and deoxy forms. The presence of two different types of protein in this oligomeric assembly allows you to have two distinct states and allows the molecule to actually function as a machine with different structures for the oxy and the deoxy forms. So I said that there's a subtle genetic difference, and it's in the gene that encodes the beta the beta chain. This is the HBB gene. There's a single nucleotide difference, single base difference, which converts the normal glutamic acid at amino acid number 6 to a valine. This GAG encodes glutamic acid, GTG encodes valine. So the consequence at the level of the protein is to introduce a valine here in the at position number 6 in the polypeptide chain of the beta subunit. What this does in terms of the surface of the protein is to introduce a hydrophobic patch. And these hydrophobic patches are water hating. And so what they tend to do is to bind to other hydrophobic patches on the surface of the protein. And that's exactly what we see in the crystal, that you have this hydrophobic patch that's been created by the mutation, another hydrophobic patch that was pre-existing on the surface of the hemoglobin tetramer. They come together in the crystal and in turn form an arrangement like this, The mutation is here at this green position, and you can see the interactions between successive heterotetramers in this assembly, generating this fiber-like appearance. And these fibers tend to clump together, and when they clump together, they distort the shape of the red blood cell, but only when when oxygen is removed, only in the deoxy state of hemoglobin S. So this is out in the periphery, in the circulation, and what happens is that this sickled red blood cell is not nearly as deformable as a normal red blood cell, and when it enters the capillary, when these deoxygenated sickled red blood cells enter capillaries, they can actually block the capillaries, and this causes pain due to lack of blood supply to the tissue that would normally be fed by the capillary, and ultimately damage to organs such as kidneys and bones, etc. So this is we go from a single base change to a conformational structural change at the level of the entire red cell, which goes on to cause vaso-occlusion, pain, and organ damage. In a little bit more detail, you can see here the space-filling rendition of the valine side chain, its interaction with another hydrophobic patch on the surface of an adjacent hemoglobin S heterotetramer. This interface is right here in this assembly that we find naturally occurring in the protein crystals themselves. One of the challenges in the early days of treating individuals with sickle cell disease is that it's not typically evident at birth. It doesn't appear until some months after birth. And the reason for that is that while the baby's in the womb, the baby actually has what's called fetal hemoglobin in its circulation. And the reason that it has fetal hemoglobin is that it has to compete effectively with the mother's hemoglobin to get oxygen. And the affinity of fetal hemoglobin for oxygen is higher than that of hemoglobin A, the normal hemoglobin that I told you about. And so this ensures that that in utero, the baby is able to get enough oxygen. After birth, the level of fetal hemoglobin, which consists of what we know as hemoglobin alpha, the chain that's in hemoglobin A, and a different beta chain, hemoglobin gamma, which is only 70% identical to hemoglobin beta. So you see after birth that the levels of hemoglobin gamma begin to decline, and the levels of hemoglobin beta begin to rise. And if this is the abnormal sickle cell version of hemoglobin beta with that valine at position 6, some months after birth, the sickling will become evident. The reason that it doesn't become evident immediately is that when you have mixtures of hemoglobin, the fetal hemoglobin, the hemoglobin F, and the sickle hemoglobin, hemoglobin S, when you have mixtures of these in the red blood cell, you don't get sickling. It's only when hemoglobin beta, the abnormal hemoglobin beta, is completely taken over that you begin to get sickling. So typically, the disease typically appears after the first 3 months of life. Now, I need to tell you one more thing about hemoglobin F. At position number 87, it has a glutamine, whereas both, this is on the alpha chain, whereas both hemoglobin, this is on the gamma chain, whereas on the hemoglobin alpha chain and the hemoglobin beta chain, both normal and abnormal, there's a threonine. And this difference becomes important a little bit later on in the presentation. So just tuck that away, that because the presence of glutamine 87 becomes important. So the question arises, of course, well, how does this change that I described, this steady decline of the gamma subunit and of the expression of the hemoglobin gamma chain versus the hemoglobin beta chain, how is this regulated in the human body? So after birth, the expression of two different proteins known as transcriptional repressors is turned on, and they bind to the promoters of both the hemoglobin gamma 2 gene and the hemoglobin gamma 1 gene. So ZBT7A binds to the promoter of the gamma 2 gene, and that effectively blocks it blocks the binding of RNA polymerase to the promoter and prevents expression of the hemoglobin gamma 2 protein and formation of hemoglobin 2 gamma messenger RNA. The same is true of BCL11A, which controls the expression of the gamma 1 hemoglobin gamma 1 gene. Eliminating this repression at either one of these positions restores fetal hemoglobin production in individuals. And there's a genetic disease known as hereditary persistence of hemoglobin F, which in which you see adults who have hemoglobin F in their circulation. And the reason for that is that there's been a disruption of this interaction between BCL11A and the promoter. The PDB contains structures of BCL11A bound to the double-stranded DNA. So that's here in red and orange. And the BCL11A protein is shown here in an all-atom representation binding to the DNA double helix. This interaction is at the atomic level, what I'm showing here schematically in this picture. Once this complex forms on the promoter of the hemoglobin gamma 1 gene, RNA polymerase cannot bind, and you cannot make the hemoglobin gamma 1 messenger RNA. So please tuck that away, because this is going to come back in the story. So we can think, based on what we know about hemoglobin S and the behavior of hemoglobin S when it's exposed to oxygen versus when it's deoxygenated. We can think about different ways of managing and possibly even curing sickle cell disease. So one thing that we can do is, and this has been done in the past, is to actually replace the blood containing hemoglobin S with blood that contains the hemoglobin beta. So that's transfusing from an individual who does not have sickle cell disease, has the normal hemoglobin A that I showed you, and simply giving them blood transfusions. Another approach that's been used is bone marrow transplantation. So that involves destroying the bone marrow of the individual with sickle cell disease using a combination of chemotherapy and radiation, and then putting bone marrow back into them, typically from a relative that does not have sickle cell disease. So this would be one of the siblings who was lucky enough that they didn't inherit either copy of the valine 6 variant of the beta chain from the parents, and doing bone marrow transplantation. And that has been successful. It's very expensive, and it carries with it a non-trivial mortality rate during the time that the patient's bone marrow has been destroyed and the new bone marrow from the donor is engrafting in the recipient's bones. Another option is to promote expression of hemoglobin F. And it was found empirically that if you treat individuals with a mild chemotherapy agent known as hydroxyurea, that you can boost fetal hemoglobin production. But that carries with it various side effects and fairly quickly fell out of favor. Another approach would be a pharmacologic intervention, and that's been successful, and I'll describe how a US FDA approved drug, Oxbryta, was discovered and tested. So this is, conventional interventions which the US FDA has approved. There are also opportunities for genetic manipulation. These are all experimental that I'll tell you about, that offer the possibility of actually curing sickle cell disease. So let's go through each of these in turn, starting with Oxbryta. So the Bay Area biotech company known as Global Blood Therapeutics was founded with the express goal of addressing the unmet medical need for sickle cell disease. A group of talented individuals at UCSD, one of whom is a member of the RCSB protein data bank team, designed a small molecule that would fit into the central cavity of the hemoglobin tetramer and increase the affinity of the tetramer for oxygen, thereby reducing sickling. The oxy form of hemoglobin S does not polymerize. They were able to do clinical trials in affected individuals and show that there was an acceptable safety profile, and show that the patients actually benefited from taking Oxbryta in terms of fewer painful sickle crises, etc. And it received US FDA approval in 2019. And PDB structure 5-echo AT3 shows the structure of the Oxbryta complex with the carbon monoxy form of hemoglobin S. Here's a chemical drawing of the drug. It binds to the N terminus of the alpha chain, and there are actually two copies of Oxbryta shown here, superimposed on one another. In any one hemoglobin tetramer, there would only be one copy bound to one of the two alpha chains. So here was the pharmacologic approach. It is working and providing value to and relief to patients with sickle cell disease here in the United States. But it would be much better if we could cure people rather than asking an individual to start taking a drug at age 3 to 6 months and be forced to take that drug for their entire life. And there are 3 different approaches that I'll tell you about that can may offer a cure to sickle cell disease. The first involves simply inserting a new gene encoding a variant of hemoglobin beta. This variant would have a glutamine at position 87. I told you about the importance of position 87 and the fact that hemoglobin S has a glutamine at this position. So the approach that has been taken experimentally, the clinical trial is currently on hold because of some quite serious adverse events that were seen in the individuals. It involves random insertion of the gene encoding hemoglobin beta with this anti-sickling variant, randomly incorporating that into the hematopoietic stem cells that have been removed from the individual, grown up in a petri dish in the lab, and put back into the individual. The challenge here is because the insertions that are mediated by the lentiviral vector are random, you can cause genetic disruptions which can go on to cause cancers, and that has happened in a number of the individuals who received this experimental therapy, and for that reason it's currently been suspended. It has this same approach, however, has been approved for treatment of beta thalassemia. This is another hemoglobinopathy, as it's called. This is work by Bluebird Bio. They received approval in Europe. And the reason that glutamine 87 is effective is that it prevents the two, even though the valine is still present in the hemoglobin S gene, the variant that's the glutamine 87 variant that is normally seen in hemoglobin F will block the productive interaction between two different copies of the hemoglobin tetramer and prevent sickling. So that's one approach. Probably not going to be the winner in the race to cure sickle cell disease. Let me just tell you very briefly about CRISPR. I'm sure that you've seen a great deal in the news about CRISPR, particularly after the Nobel Prize in Physiology or Medicine was awarded for this work. I think it was last year. So CRISPR stands for clustered regularly interspaced short palindromic repeats. And there is an enzyme known as Cas9, which is part of a bacterial defense system, which can be repurposed to act as a pair of genetic scissors. It can also be repurposed to insert particular DNA sequences into a genome at a defined position, or change DNA sequences in a genome at a particular position. The structural biology of CRISPR is very, very well understood because of the work of Jennifer Doudna and others. Jennifer was one of the co-recipients of the Nobel Prize. And it is a combination of, the system is a combination of protein, RNA, and DNA, and it's the guide RNA that provides the selectivity in terms of the interactions with particular parts of the genome. So one approach for curing would be to promote expression of hemoglobin F and essentially simulate or create what's seen in individuals with hereditary persistence of fetal hemoglobin. And this will be done by removing the binding site in the promoter for the BCL11A transcriptional repressor. So the idea is that using the Cas9 system with the appropriate guide RNA, it will be possible to make a change to the DNA, either by making an insertion or by making a deletion. Both strategies could work, and it would have the effect of abolishing the formation of this complex of the transcriptional repressor and the promoter DNA that I showed you previously. There's another option that also involving CRISPR that would allow for correction of the hemoglobin S mutation directly. And I use the word correction in quotation marks because it's a change. It's not a perfect correction. So when CRISPR-Cas9 forms a complex with another enzyme known as adenosine deaminase, you can change in a particular position in a genome an AT base pair to a GC base pair. So you recall that I told you that there is in the sickle cell, in hemoglobin S at the codon that encodes valine number 6, there's an abnormal TA base pair. What one would like to do is to actually fully correct and change this TA base pair into an AT. That's not possible with the adenosine deaminase CRISPR-Cas9 system, but what is possible, at least in principle, would be to use the adenosine base editing and to put a CG base pair here instead of a TA. And this will give rise to what's known as hemoglobin G, which when deoxygenated, will not sickle and will leave the red blood cell looking perfectly normal. So this involves this conversion at codon number 6 from GTG to GAG will generate what's known as hemoglobin gene macassar, which is a benign variant of the hemoglobin S. And the machine that would do this has actually been studied using X-ray crystallography by Jennifer Doudna and her colleagues. And what you can see is you've got Cas9, you've got the guide RNA, you've got the DNA, and then you have the adenine deaminase that's going to do chemistry on the genome and make that single base change. And if this happens only in the position of valine 6 in the gene that's encoding the defective form of the beta chain of hemoglobin, and making no other changes anywhere else in the body, then you could potentially cure individuals of sickle cell disease. I say if it only happens, because there are still concerns that these base changes may occur elsewhere in the genome, and there's always the possibility of an unwanted effect from doing so. So to summarize very quickly, I've told you about the Protein Data Bank, and I've specifically highlighted not only what we're doing in terms of global research and education, but what we're doing for COVID-19 at the US-funded RCSB Protein Data Bank. And then I've described the history of sickle cell disease, the first molecular disease, shown you at the atomic level the molecular basis of the disease, and also shown you at the atomic level how the disease is currently being managed, and opportunities for genetic intervention that could create cures. So with that, I need to acknowledge the enormous number of talented people who work at the RCSB Protein Data Bank, allowing me to stand at the tip of the spear and tell you this story. I want to specifically acknowledge Dr. Shuchi Dutta, who helped me prepare all the sickle cell disease related slides from work that she's been doing, funded by the National Science Foundation. But the health and vitality of the entire archive and our data delivery system depends on every one of the individuals depicted here. Before I advertise some job openings, both at Rutgers and on the West Coast at UCSD, I need to acknowledge our funders, the National Science Foundation, 3 different institutes at the National Institutes of Health and the US Department of Energy. I need to thank our hosts at Rutgers, UC San Diego, and UC San Francisco, and also acknowledge the contributions that are made by our partners in the Worldwide Protein Data Bank, and to thank you for your attention. And I'll close and just leave this slide up during the Q&A that advertises software developer positions both at Rutgers and UCSD, and a postdoctoral researcher position at UCSD. Any of you who are interested in becoming members of the RCSB PDB team could go to our jobs page, www.rcsb.org/pages/jobs, where you can find all of the details about the opportunities to become part of this team that's benefiting so many people around the world on a daily basis by converting global data into global knowledge for 3D structures, and advancing our understanding of fundamental biology, biomedicine, bioenergy, biotechnology, and energy sciences. And with that, I will stop and thank you, and I'll be happy to take questions for the remaining time.
Hello.
Hello.
Hi, this is Charmaine. First of all, great talk. I work on sickle cell disease, so I was pretty excited to hear your talk and to see all the details. I'm sorry that I came in a little bit late, but my question is, what is the difference between the fetal hemoglobin versus the adult in terms of the way it can take up oxygen and deliver oxygen to the tissues? Are there any differences?
Yeah, so interesting question, Charmaine. And this is actually the subject of a a Molecule of the Month article, which if it hasn't been published soon, it will be, hasn't been published, it will be published soon on our pdb101.rcsb.org website. And the Christine Zardecki, the Deputy Director of RCSB PDB, is providing the link in the chat. So hemoglobin F has a slightly different central cavity. That central cavity is unable to productively bind 2,3-diphosphoglycerate, which is a chemical that's present in our tissues that induces the hemoglobin to give up the oxygen. And that can't bind to hemoglobin F. It binds to the mother's hemoglobin in the placenta and induces the mother's hemoglobin to give up the oxygen. And then hemoglobin F will bind, under those conditions, will bind oxygen more avidly and will pick up the oxygen from the mother's hemoglobin and then transport it to the developing fetus and provide all the oxygen nutrient that's required. You can, I think, see very clearly that if a baby had hemoglobin A and the mother had hemoglobin A, it would be impossible for the baby to actually compete effectively for the oxygen from the mother's blood. So the baby's blood has to have a higher affinity for oxygen, and it does so through sort of modest differences in the amino acid sequence that mean that it doesn't bind 2,3-diphosphoglycerate, and hence is able to bind oxygen after the mother's hemoglobin has bound 2,3-diphosphoglycerate and given up the O2 molecule. So that's the explanation.
Okay, great. That makes sense. That's what I speculate. I have a second question.
Okay.
It's regarding the Bluebird trial. I haven't really been following it closely, but from what I heard, it's not the virus that, so those patients that received the gene therapy got AML and
yeah, so there were, I think there was both myelodysplastic syndrome and acute myeloid leukemia were seen in individuals who had received the lentivirus.
So what, but I think the company is saying it's not because of the therapy, right? That it's the patients themselves were prone to that.
Right now the trial is suspended, I believe. And it is incumbent on the company to prove, in order to be able to continue the trial, to prove that this is unrelated to the treatment. Now, we know that that same company has been successful with the lentivirus that has been used to treat transfusion-dependent beta thalassemia, so-called TDT. There's reason to believe that it should have worked in individuals with sickle cell disease, and it may simply be bad luck for the company that two participants in the clinical trial developed these quite rare hematologic malignancies. But these hematologic malignancies are quite rare, and simply on a roll of the dice, you wouldn't expect the individuals enrolling in the trial to develop cancer, because it's just not something that happens very often. So I think the jury is still out there.
Okay, my third question is about the CRISPR-Cas9. I've been seeing a lot of papers where they target the induction of the fetal hemoglobin instead of correcting the mutation for the HbS.
Yeah.
And I'm wondering if that I didn't completely understand why that strategy, why they're choosing that strategy better. Is it because it's harder to target to correct the SBS?
I think it's much harder to do a single base correction than to go in and do a strategic deletion or insertion in a promoter. Okay. I think that's the reason that they're trying it. And there is a genetic condition, the hereditary persistence of fetal hemoglobin, that is observed in some individuals, and they those those individuals are not sick. They are perfectly happy with fetal hemoglobin on board. And when fetal hemoglobin is mixed with the sickle hemoglobin inside the red blood cell, it prevents formation of the sickle fiber and prevents sickling.
And thank you so much, Stephen. That was amazing.
Well, thank you. I appreciate the opportunity to give this talk. It's the first time I've ever given it. This is new material for me, and I'm very glad I had the opportunity to give the presentation to Venture Cafe this afternoon.
Awesome. Well,
Very
thank
fascinating.
you so much.
Thank you.
And
Thank you.